Okay, welcome to the fifth video. This one's the second one with biodiversity in ecosystems. And basically we're going to look at a bit about the influence of nature and humans on biodiversity, and then about how we measure biodiversity. Nature is all about increasing biodiversity as much as it can in a particular area. What you'll tend to find is that most habitats start off in a particularly harsh environment, whether it's bare rock from plates of the earth rising up, maybe the earth rising out of the ocean, and therefore rocks rising out of the ocean, and therefore creating desert islands, seas receding and creating a sandy beach. But all of these don't have many organisms there, or any in some cases. Are very harsh conditions because there's no nutrients, and as a result it's very difficult for plants and animals to live there. Biodiversity is low. Now what will happen is some particularly well adapted organisms that are really good at surviving in these difficult conditions, such as marram grasses, which are often found growing on dunes. These will come and grow in the area because they're very good at getting what little nutrients there are there, and they also are good at getting carbon dioxide from the air, changing it into nutrients through photosynthesis. That's the plants by which plants produce food using light. So then what will happen is all the nutrients that make up that hardy organism, when it dies and decomposes, it then becomes available to others in the soil. So essentially what's happening is its carbon dioxide is being drawn into the soil and through the plant. And as the conditions become, there's more and more carbon and nutrients in the soil, more complex species of plants can grow, shrubs, herbaceous plants, and then with different varieties and a higher biodiversity in plants, then greater varieties of insects and other small animals can grow, and gradually then predators which feed off them will come to the area as plants become even more complex. There's shelter and nesting space, and eventually you come to quite a complicated high biodiversity environment where there are plenty of nutrients. Humans, on the other hand, tend to decrease biodiversity in a particular area, and it's only recently we've been noticing the importance of it and trying to counter this. One good example of this is monoculture, where we grow one crop over a very wide area, acres and acres, with just say rows and rows of cabbages. Now because of this, you've only got one type of plant. There's not going to be as many organisms adapted to survive and feed off just one plant. So therefore you're going to have fewer species feeding off the plants, and as a result then the species of organisms that feed on those consumers, there'll be less of those, and biodiversity decreases. You can couple this then with the fact that herbicides are used to kill weeds and insecticides are used to kill pests. And okay, we might not consider weeds and pests as species we want in a particular environment, that they've got this bad image to them, but nevertheless they are species that are naturally in that environment, and they are part of biodiversity. There's other ways in which we influence biodiversity, for example by tarmacking and cementing areas. You look at a car park, and that is one massive amount of tarmac. How many plants do you see growing through there? None, unless there are a few cracks where the most hardier plants are coming through. So there's another influence of humans in decreasing biodiversity to virtually non-existent. Now, to see the effect that we're having on biodiversity and hopefully to try and improve it, we've got to be able to monitor it and measure the biodiversity. And there are three techniques that we can use. One of them uses a small square called a quadrat. It's a wooden frame basically, and what we do is we take a map of the habitat that we're trying to monitor and chop it up into put a grid over it and give a number to each of those squares. We then use a computer to randomly generate a number, and we place the quadrat on the ground in that correct location that the map is telling us. And we can then look at the species within the quadrat, count them up, and we do that over a repeated number of times, and then we can work out the average number of species there are, organisms there are of a particular species. This has got two problems with it. Firstly, if the species or the different types of species aren't evenly dispersed, then you're going to have problems. For instance, with a pond, you can have the bank where there's grass, near the pond where there's the edge of the water where there's reeds, and then in the actual pond itself, well, you only have lilies. Now, your problem there is, what if the coordinates we pick to test and to measure are only on the bank where the grasses are and in the pond where the lilies are? That would be, the statistics that we draw up from that would be telling us that, well, there's no reeds whatsoever in that habitat. All because our coordinates missed the ones that were around the edge of the pond. So if we can only use a quadrat if the organism we're measuring is evenly dispersed. That's such as daisies in a garden. The other problem, of course, is that if the species we're trying to measure is motile, it moves around, we're going to have problems again, because if you put the quadrat on top of it, of course, it's going to run away. It's usually very good for plants because they can't move, or sessile organisms that can hardly move at all, such as snails and slugs. They're not going to move away by the time you've got enough time to put the quadrat down and count them. So if it's not evenly dispersed, then we don't use a quadrat, we use what we call a belt transect. And that's a row of quadrats going across the habitat. Now you can, depends how frequently you want to put them, you can put a constant row of quadrats, or you can put a quadrat along this belt of quadrats every 4 meters or so. But obviously it's a measure of how much time you have, how much labour you have. Obviously the more of them you do, then the more reliable your results will be. But it takes a lot longer to actually collect tons and tons of data. Now, because of collecting them as a belt transect, we can see then that how the organisms are dispersed and how the population numbers change over the particular area or throughout the area. Again, though, this has the problem with the idea of it only works with sessile things, because again, it's simply placing quadrats in a line. So anything that's motile is going to just move away and run off. So we've also got the capture, mark, recapture, release method. Now, capture, mark, release, recapture. What that does is, what we do is we set up a small trap which doesn't harm the actual creatures. For instance, with some sort of insect, you can put down small jars and they fall into the jar. You then count the number that you've collected, put a little mark on them, something which won't increase the likelihood that they'll be picked off by predators, or it won't damage them anyway, and will over time be removed. And then you release them. You reset the trap, same amount of time that you left for the first capture, and then you capture again and you count the number in your second sample that was still carrying the mark, that essentially you've recaptured. And then you can calculate the population size by using the equation that you can see in the PowerPoint. Now, the point of this complicated procedure, why not just capture them and count them? Well, your problem with that is that some of the species, because they're motile, some of the ones that you capture will simply be passing through the area. They're not part of the population in the habitat. They're merely migrating, perhaps from one area to another because the food is lower in one area. Perhaps there's been just after a breeding season, there's larger numbers of them, so they spread off to find their own homes. But you can't count those ones as part of the population. And capture, mark, recapture allows us to take that into account, because the ones you recapture and are marked will be the ones that tend to be in that population, in that habitat over a long period of time.